Good morning. I welcome everyone to this meeting, all the panelists, speakers, and delegates to the session on emerging pollutants and wastewater of UNESCO IWRA third conference. So I'll be moderating this session. I'm Dr. Suryakanta Acharya. I'm the founder of PW Clinic. It's an NGO working on health and climate change. I'm a member of IWRA Water Quality Task Force and also a scientific committee member of this conference. For this session, we have co-moderator Dahlia Sabri from Egypt. She's working as Deputy Resident Engineer in KO International Consultant. She'll be managing the Q&A section. And for this session, the reporter will be Nusrat Baruch from Pakistan. She will take a summary notes for all presentations to be presented at the end of the day. So just some housekeeping rules. Just to remind all our speakers, this session is being recorded. We invite you to submit your questions in English via Q&A button. You can find this button at the bottom of your screen. We'll address your question as much as possible after the talk or in the Q&A section. You'll only see each question once it is answered either verbally or in writing, when they'll be displayed for everyone to see. As we have a huge audience, if you don't have time to answer all the questions we receive in this session, we'll aim to collect written responses from the presenters to place on the conference website following the event. You will also have the option of switching on the automatic subtitles at the bottom of your screen if you have any difficulty in hearing. If you experience any technical problem, please Google Zoom Help Center or send an email to online.conference@iwra.org for assistance. Please note that the chat box will be disabled during the session. However, we ask you to keep an eye out for any useful messages that may be shared in the inbox from the organizer and moderators. So we have a keynote presentation which will be by keynote speaker, Professor Gordon McKay. He is a professor at the Division of Sustainable Development, College of Science and Engineering with Hamad bin Khalifa University, Qatar. His research area include wastewater treatment, absorption and ozonation for emerging pollutants, energy production and efficiency, and value-added products from waste baccalaureate for agriculture. He was awarded with the DSc in 1992 for research using absorption in wastewater treatment, and he had published over 600 referred research journal papers. Professor Gordon, back to you.
Thank you very much for this opportunity to present our research on emerging pollutants, pitfalls in their removal, a case study. I'm here with my co-author, Dr. Jaya Sathasavam, and we'll commence. Next, please. Okay, so the US EPA has defined emerging pollutants as new chemicals without much regulatory status that impact on human and health and are generally poorly understood. Going down to the bottom bullet point there, we look particularly in this presentation at pharmaceuticals, personal care products, and these are widely used all over the world with enormous diversity and in enormous numbers. Next, please. Here we can see this enormous diversity. On the right-hand side, we have two pictures of pharmaceuticals and then the healthcare products, just showing the large range of common materials available. More than 3,000 pharmaceuticals used as medicines in so many applications, anesthetics, antibiotics, etc. Some are EDCs or endocrine disrupting compounds, which of course are very dangerous. We have polyfluoroalkyls, veterinary pharmaceuticals, micro nanoplastics, pesticides and herbicides, to mention just a few. Next, please. In terms of pharmaceuticals, these are taken by humans and discharged into wastewaters from human bodies after metabolism or by direct disposal in toilets. Personal care products are discharged. These are the health products like during showering and bathing. Other common routes: inappropriate disposal, landfill leaching, agricultural runoff, rainwater, livestock and veterinary waste. The PPCPs and DDCs are discharged from production industries, domestic and industrial wastewater effluents, hospital waste and wastewaters. Next, please. Here we see it in a diagrammatic format. The left-hand diagram on the screen, if we look at the left-hand side of that, we see the discharge routes, so the fake routes, direct disposal, industrial effluents, untreated waste releases. And these then move across from the left-hand side to the right-hand side and will be discharged in the form of sewage, surface water, seawater and groundwater. On the right-hand side, we look at the particular case of the sewage treatment techniques. So in the red block, we can see again the range of materials that we're talking about, and then they go into treatment and come out as perhaps biosolids for soil application or effluent for water applications. And within that framework, then as they're sort of being discharged, we can see they can undergo metabolism, bioaccumulation in cells, absorption and adsorption, and the degradation into another whole range of byproducts. Next, please. The adverse effects, we've really covered these, but they can enter the food chain, enter environmental species. We've already said some are endocrine disrupting compounds, the EDCs, found in pesticides, steroids, metalloids, and perchlorates. They can cause hormonal problems because they're classified as estrogenic, androgenic, or thyroidal. And they also cause feminization and anomalies in reproductive organs, particularly in aquatic creatures such as fish, algae, frogs, etc. Next one, please. Here we just take one example before we go into the treatment studies. Metformin, consumed by diabetes patients, is one of the most found pharmaceuticals in treated wastewater effluents. It's a non-hormonal pharmaceutical that can cause problems naturally in the hormones. It can cause agonistic issues in cells and antagonistic as a hormone blocker. And that is, as I say, just one example of our pharmaceuticals. Next, please. Okay, just summarizing now the adverse effects, toxicity. We have effects such as neurotoxicity, genotoxicity, carcinogenicity. The environmental impacts incorporate water and soil pollution, harming wildlife and reducing biodiversity. Antibiotic resistance has been identified, making it more difficult to treat infections and can lead to the spread of antibiotic-resistant bacteria. Next, please. Moving into the study now. So we have to remove these materials. We need to design treatment processes to remove them, and the problems are now summarized. The emerging pollutants, many of them cannot be removed completely by the conventional wastewater treatment plants. Another issue is there's no target compounds. There are several compounds in the wastewaters with different properties, so it makes it difficult. Which process shall we select? There are hundreds of different structures and end groups. The emerging pollutants are present in low concentrations in large volumes of wastewaters. And this, of course, is an issue in itself. It's difficult. They are present as multi-component systems. This is really going back to bullet point 2. There are many compounds there with many different properties. And we come down to the bottom statement on this PowerPoint. Therefore, selecting and designing a single-stage treatment system for an emerging pollutant wastewater source will not be sufficient. We're going to have to look probably, as we develop these treatment systems, at a train of processes to do the treatment. Next, please. Okay, so we took on a case study to look at 2 of these to try to see and compare different treatment systems and how they can be done. If we move to the right-hand figure, the 2 compounds selected by sort of talking to people and industry, as just to pick two common ones, was gemfibrozil, which is a lipid regulator, and ibuprofen, which of course is the famous analgesic. Chemical formulae are also given in that table, and we see that they are phytotoxic to carrots, tomatoes, lettuce, and other vegetables. They affect sea creatures, and they affect humans. Next, please. So this is the program of study. If we look at the little block schematic at the side, what we adopted, we want to basically, in Qatar, like much of the Middle East, we have a huge water shortage problem, and we pay huge amounts of money for desalination processes. So we're trying to maximize treated sewage effluent for agricultural reuse. To do this, we want to remove pharmaceuticals. So this case study will remove ibuprofen and gemfibrozil in the single system, so that is each in water, and then the two combined in the treated sewage effluent. We studied 5 processes: chlorination, ozonation, ozone with hydrogen peroxide, adsorption, and catalytic ozonation. And we evaluated the efficiencies of these under different experimental conditions. We also, in some of the cases, monitored the dissolved organic carbon and nutrients to see what other impacts were coming into play in the TSE. We did a little economic evaluation as well. Next, please. So we start into these processes now, and the first we were going to look at, we decided to take on adsorption. So we thought we'd produce our own activated carbon from a local waste. And of course, in the Middle East, we produce beautiful dates. So there's a big industry been developing over the past 10 years or so in producing seedless date products like jams and syrups, etc. So once the dates have been eaten, or let's say the seed removed, in these processing plants, there are quite large quantities available for testing as the date pits or the date stones. And that's the second block diagram shown there. What we did with this was we shred them, treated them with phosphoric acid, and then heated them up to 550 degrees centigrade for 2 hours to produce an activated carbon. And this, let's say, we will use for adsorption. So next one, please. This shows the results of the adsorption study, putting the adsorption into the TSE that's got ibuprofen and gemfibrozil in. And this is the figure. We'll just look at the bar chart there. The left-hand scale is the percentage removal from 0% to 100%, and the right-hand scale is the carbon dosage. So if we look at the first bar chart, the left-hand figure, we've 4 bar charts, 4 bars there. Right. Blue and red are ibuprofen, single and binary, and the green and purple are gemfibrozil, again, single and binary. Low concentration of carbon, so not full removal. But then if we move across to the right, we look at the last 3 figures, we see we've reached an optimum dosage of carbon, and we can remove gemfibrozil 100%, which is great news. But whatever we then tried with ibuprofen, it's difficult to get above 80 or 82%. So we were meeting a little problem there. So we move on and try another technique. Next, please. Ozonation only. So the first of those figures on the left shows the removal of gemfibrozil. The blue star is at lower concentration, which is like over 90%, but at a higher ozone dosage, we can get complete removal again of gemfibrozil. So we're not having too many problems with gemfibrozil, but when we flick over to the right-hand diagram for ibuprofen, different concentrations, binary and single, even at the highest level, the top 2 graphs in the red and the circles, we cannot go much above 80%. So again, ozone has limited effect on its own. Next one, please. Okay, so this was good news, catalytic ozonation. So we've got the ozonation and we add our catalyst, which in fact is the activated carbon that we produced before. And we did 10 batch samples using the same carbon. And so there's 10 sets of results in that left-hand bar chart. And you can see most of them are close to 100% with this combined treatment process. The dark blue is our ibuprofen, and the lighter blue is gemfibrozil. Gemfibrozil, again, in every case is 100%. And even after 10 batch studies, we're still getting above 90% for our friend ibuprofen. So this combination, catalytic ozonation, is producing very good results for us. Just moving down there quickly, you see the light blue, that's the third bar chart, the third bar, that is decreasing, right? Now, this is dissolved organic carbon, really representative of degradation products. So there is a hint that the carbon is becoming more and more loaded. At the bottom, there's some good news. In the presence of TSE, of course, there's phosphate and nitrate, which can act as fertilizers and nutrients for agricultural irrigation purposes, and those are hardly removed at all in these processes. So they will still be there in the treated sewage effluent if we can use it for agricultural purposes. Moving over to the summary, the right-hand diagram, the 5 processes are shown there together: chlorination, ozonation, ozone plus H2O2, adsorption, and catalytic ozonation. You can see chlorination doesn't give great results, right? Fairly low removals, but once we go to ozonation, which we've already seen, we can get 100% gemfibrozil and over 80% ibuprofen. Ozone and H2O2 didn't give much difference to the ozonation only. So you can see very similar results. The adsorption results, again, which we've studied, 100% and maybe heading towards 90%, but not complete removal of ibuprofen. And then the final one, good news, catalytic ozonation gives 100% for both. The little line going through those are the relative costs of operating the different plants. Next one, please. This is the summary. So basically, we're summarizing by saying that the tertiary treatment technologies are giving us high removal, particularly catalytic ozonation giving us 100% removal. Expensive, but successful. Future work, we looked at oxidation byproducts, as we saw in the previous slide, assessment of bromate formation, that could give further problems, and then we want to try a pilot scale test at the local sewage plant. Ladies and gentlemen, thank you very much indeed.
Thank you, Professor Gordon, for the excellent presentation. And now I invite my co-moderator Dahlia to speak about herself and a bit about the Q&A management.
Thank you, Surya, and thank you, Gordon. I have one question. One of the attendees asking about what is the EDCs, so maybe you can just shed a little bit light on how it affects the hormones. And
okay, I'm not a medical expert. Of course, the EDC stands for endocrine disrupting compounds, and it's been discovered that there are quite a lot of chemicals, particularly coming through the pharmaceutical range of chemicals and pesticides that do affect sort of our endocrine, our metabolistic processes. But as a chemical engineer, I'm not a doctor, so I can only say that they need to be removed from the waters. Yeah.
Another question is, considering there is no regulation for the EPs, how do we decide to design the wastewater treatment system?
Yeah, well, that was, in very much like the whole purpose of what we were trying to do, like select a couple, not randomly, but by talking to people, a couple of materials that occurred quite regularly in the effluents, and then see, okay, let's try different treatment processes and how effective they are. And we saw that, like for one compound, gemfibrozil, almost any treatment process will take it out quite successfully. We came back to ibuprofen, which of course is a very commonly used medicine. And that guy was giving us a lot of problems all the way through until we combined two processes, the catalytic ozonation, the ozonation with activated carbon. So I think it's an area, well, I think everybody here is probably biased, but it's an area where there's a huge amount of research potential to be done to look at this and look at the different compounds, because there are so many different compounds, so many different chemical structures. What sort of systems are needed? And as I said, I started at the beginning of the treatment system. I think we will need a train to treat this type of sewage effluent with maybe 50, sometimes 100 different compounds in. We probably need a train of 3 or 4 treatment processes to be successful eventually.
Thank you.
Thank you.
I think this question also is related to the previous question regarding the ibuprofen. Were there test results about possibly harmful residues emerging from the treatment process, like oxidation?
Okay, I'm possibly going back to, did you know this about the breakdown products? We were only measuring, we were only looking at the dissolved organic carbon. And so that was an indicator that there are degradation products there. We have started to look at the brominated compounds, and we find that they are enhanced as well. So I think the second phase of these treatment processes will be to look at the breakdown products and check to what extent they are removed. Because some will certainly, we didn't look at too many types, but certainly some will be more toxic and some less toxic than the original parent compound. Thank you. Thank
you. I have this interesting question. It says the cost and availability of land in urban areas is a basic impediment, while energy cost is very high in land. So can we reduce the pollution load?
Not fully sure what is meant by that. I mean, the process reduces the contaminants, particularly with the treated sewage effluent. Our target in Qatar, and it says representative, a lot of the Middle East, was to try to reuse like the sewage effluent, but for a more practical purpose in agriculture. And of course, if we're going to pump out the normal material with all these emerging pollutants in them, they will accumulate and be absorbed by the plants, or the fodder, the crops, which will then be eaten by the cattle and come back through the food chain. So, I think we are reducing the pollutants, but at the moment we're using advanced treatment processes, which we've already said are moderately expensive. You go back to the cheaper one, the chlorination, which is the cheapest, because it's well established, dumping in hypochlorites and things like that. But it had poor removal properties, and also when you put chlorine in, you're going to produce chlorinated products, which is not a good idea. So at the moment, from our results, we're looking at fairly expensive treatment processes, ozonation, catalytic ozonation, production of active carbons. Yeah. So we want to reduce the polluting load, but it's a little bit expensive at present with those technologies. Thanks. Yeah.
Thank you. Well, I have a question. Somebody wants you to elaborate a little bit on other challenges in wastewater reuse besides the EDCs.
Besides?
The EDCs, endocrine.
Oh, besides the EDCs. Oh, there's so many there. There are so many, and I mean, moving into other areas. I mean, one area that's been studied for a long time is like removal of, let me think, dyes from textile effluents, and there was more researchers going into that. They're finding that a lot of these particularly have toxicity properties, carcinogens, etc., etc. So the applications may become a little bit wider in that respect, in treating waters. I mean, many years ago, we did a project with Ozonation back in Ireland, and we were trying to remove humic acids, you know, because of the humic acid stain coming through into the drinking waters. And that was moderately successful, but quite tough. And again, because the concentration is important when you're using ozonation. When you go to having to put in high doses of ozone, it does become pretty expensive. You know, but yeah, I think there's lots more sources of emerging pollutants going to appear in the future, as a summary, yes.
Thank you very much,
Thanks.
Gordon. And for the sake of time, I think we have a couple of questions. If you can please type your answers in the chat Q&A box.
Yes, I'll do my best. Thank you very much indeed.
Thank you. Back to you, Surya.
Thank you, Professor Gordon and Dahlia. For rest of the presentations, we'll answer questions at the end of the session. So our next presenter is Ubhat Ali from Indian Institute of Technology, Jammu. He'll be presenting from India. Ubhat Ali.
Thank you, sir. Good afternoon, everyone. My name is Ubhat Ali, and I am currently pursuing my PhD in Environmental Engineering from Indian Institute of Technology, Jammu. I'm humbled to receive this opportunity to be able to present in this conference. The topic of my presentation would be addressing emerging contaminant removal in hospital wastewater treatment using algal-bacterial consortia. Next slide, please. The present mechanism of wastewater treatment that is adopted mostly throughout the world is a centralized scheme of treatment, wherein centralized wastewater treatment plants exist, which are made to treat the wastewater of the entire community. That emphasizes the parts of the community like the domestic parts, the industrial parts, and the hospital parts of the community in a single wastewater treatment plant. What happens by this is that loads of epidemiological vectors are present loaded into the wastewater treatment systems, and these wastewater treatment systems are only capable of removing the nutrient pollutants, and that too to a very limited degree of efficiency. So what is best for this current scenario where the world is moving towards a tremendous water scarcity along with a high rise of pollution throughout the world? The best treatment technology to be adopted nowadays is a decentralized or a modular wastewater treatment mechanism. These decentralized or modular treatment mechanisms could serve as point-of-care systems. They could be adopted in just in a domestic community or various societies, or to specific parts of the community wherein they would address to the exact emerging contaminant or the exact kind of nutrient pollutants that are present in the wastewater and could be helpful to remove the same. Next slide, please. So the country that we're addressing currently, I'm from India, and India is facing huge pollution throughout the country as such. So hospital wastewater nowadays, especially because of the COVID-19 pandemic, is loaded with epidemiological vectors. And when it is mixed with municipal sewage, what renders is that most of the emerging contaminants, which are dangerous to the atmosphere and environment in general, are released as such into the water streams or into the atmosphere. What happens after that is that these pollutants would, over the period of time, not now, but over the period of time, would accumulate within these water bodies or within the land and wherever, and they would probably lead to the next pandemic that we could see over a period of time. The daily wastewater discharge from hospitals ranges from about 400 to 1200 liters per capita per day throughout the world. The hospitals discharge persistent organic pollutants like pharmaceutical compounds, like chemical compounds, like surfactants, and most of all, antibiotic-resistant bacteria that are now developing, which would require a fourth generation of drugs that has not even being generated to date as yet the stocks are going on, but it is not being generated. So the volume of wastewater that is generated from hospitals is tremendous on itself, and it also depends upon the size and type of patients, the patient intake capacity, the technical facilities, the miscellaneous services provided to the patients, and the mixing of sewage and hospital wastewater would obviously choke the treatment units of the centralized treatment mechanism. Thereby, a deep need is there to move to modular systems of treatment. Next slide, please. Now, this is a pictorial representation of what the world is currently going through. The research gaps in our lab that we are going to address would be the emerging contaminants, which are predominantly a great research gap that exists because very less work is going on throughout the world, and not many removal mechanisms exist as such which are able to remove these contaminants. Microbial biofilms are generally being used throughout the treatment mechanisms that are being used. Centralized systems are being used, and the aeration requirements in the treatment plants or the centralized treatment plant is tremendous, which also incurs a huge capital cost to, from the budget. So what in our lab we are suggesting is that we will develop a technology using algae. The algae which are intrinsically present in wastewater would be used, and they would help to remove the conventional contaminants along with the emerging contaminants from the wastewater, and the process overall would be known as the alga MBBR. So next slide, please. We primarily are working on three hypotheses as such. The first hypothesis being the intrinsic symbiotic relationship between the algae and the bacteria that are functioning. Secondly, the algae as such are able to bioaccumulate, they're able to biodisintegrate and bioabsorb the contaminants that are present in wastewater, and they are green in nature. That is, they're intrinsically present, and we don't have to put in any effort or any monetary cost in the cultivation of algae. We just have to provide certain favorable environmental conditions and they would grow. And by the end of the day, what is the result? We are getting wastewater with a relatively lower contaminant load, and which could be released into the atmosphere as such, but would be less poisonous or less contaminant prone. Thirdly, the algae, being autotrophic in nature, are able to generate oxygen of their own. And this oxygen, what would it do? It would help to reduce the need for aeration, and the algae would then adhere to the MBBR or MBBR carriers that are present within the system, and they would economize the MBBR treatment technology. Next slide, please. The objectives of the research study is we aim to reduce the aeration requirement as discussed. We aim to receive appreciable removal of antibiotics, surfactants, and phenols, which are generally present in the hospital wastewater. We are going to study the removal rates of the nutrient as well as the emerging contaminants using the novel algal-bacterial consortia. We would conduct the ecotoxicological studies such that the algae would be able to remove certain antibiotic parent compounds from the wastewater, but then the certain daughter compounds would be generated from these the removed compounds. These daughter compounds need also to be assessed for ecotoxicological studies such that they should not be toxic to the environment after releasing this wastewater to the atmosphere. Finally, what we are going to propose is a modular, sustainable, economic, flexible, and a green approach for hospital wastewater treatment. Next slide, please, sir. The methodology that we're adopting in our lab is first we're culturing the algae from the wastewater that are present intrinsically. The wastewater we are obtaining is from the wastewater plant that is present intrinsically within the campus. Then we are conducting a central composite design on the wastewater samples that we are generating in various flasks. From these flasks, the best of the removal rates are achieved from some of the flasks, and some of the flasks are not able to perform up to our expectations. The flasks which are the best performing are then bio-augmented into the ABC MBBR reactor that are present, that is seen in the batch operation figure. Then this ABC MBBR reactor is studied in comparison to the conventional MBBR reactor, which is just focusing on the treatment using the bacterial biofilm as such. So next slide, please. Further, these carriers that are present within the both of the reactors, what happens in them is that they develop a microbial biofilm on them. This biofilm is essentially responsible for the removal of the nutrients as well as the emerging contaminants. But we know that the bacterial biofilm is a delicate biofilm, and it is not very well capable of removing the emerging contaminants especially, and the nutrient contaminant removal is also to a very less limit. So what happens is that when we're using the ABC microbial biofilm, the algal-bacterial consortia, they are capable of removing the nutrient contaminants to a much better level as compared to this. And we are studying a comparative relation between the two. GPS6 software, Super Pro and CMAPO would be used for the validation, the techno-economical analysis, and the life cycle assessment of the same. Next slide, please, sir. The preliminary results that we're able to obtain from the batch studies that we're able to conduct in the lab is that we have been able to assess the ammonia kinetics of the same. Although a comparative result has not been shown over here, but the ABC, as is seen, is able to remove up to 50 ppm of ammonia within a span of just 8 hours from the same. However, the bacterial biofilm would require a hydraulic retention time of approximately 16 hours from the same. The surfactant and the phenol removal from the reactors is also found to be of an appreciable limit over a time period of 2 days. The microscopic image of the culture has also been attached. This has been done such that to show you the predominance of the algal community as is seen from the green color of the image as such. Next slide, please, sir. The preliminary conclusions from the study are thereby suggesting us to move further with the same. We are able to remove the emerging contaminants along with the nutrient contaminants in a much lower HRT as compared to the same, and 70% of removal and more than 80% of COD removal has been observed. Further, what we are going to do, we are going to run this same cycle at a pilot scale in a continuous mode, and we would be observing the removal rates of both the emerging and conventional contaminants. And further, we hope that we will be able to achieve a novel mechanism that is modular in approach and is able to render the hospital wastewater free from these contaminants. Next slide, please. Thank you, everyone. Thank you. And sorry for exceeding the time. Thank you so much.
Thank you, Ubhat.
Thank you, sir.
So our next presenter will be Obaid Alharbi from National Center for Water Technologies, presenting from Saudi Arabia.
Okay, thank you. Hi, everyone, and welcome to my presentation. I'd like first of all to thank organizer to give me this opportunity to present this study at this conference. So my presentation will be about the removal efficiency of pharmaceutical residues from 3 wastewater treatment plants in Saudi Arabia and their potential for pollution of groundwater. Next, please. As you may know, pharmaceutical has been reported and detected in different infinite metrics such as groundwater. Groundwater is, as you know, Saudi Arabia is located in a very dry climate zone and has very low rainfall. And because of the scarcity of fresh water, absence of scarcity of fresh water, wastewater is used for irrigation and artificial recharge. So protecting this very valuable source of natural water is very important issue for Saudi Arabia. Next, please. Methodology, we investigated 16 compounds. We collected about 144 samples from 3 different wastewater treatment plants, and we be able to detect 11 and 5 compounds in effluent and effluent of these different plants. Next, please. So the result turn out like for the average removal efficiency of these pharmaceutical through these different plants was almost high, almost above 80%, except 2 antibiotic compounds, which was ciprofloxacin and ofloxacin, especially in the small scale plant. And as you can see in this bar chart, baclofen for the first time worldwide has been detected, and its removal efficiency estimated as well through these different treatment plants. Next, please. So as well, we investigate the leaching behavior of these pharmaceutical through the soil column at lab scale. So by applying artificial rain for almost 48 hours, which is equal to 251 rainfall, and then we analyze the soil, and the leaches came through this soil as well. Next, please. So by applying this artificial rain in this soil column, then we'll be able to study the behavior of these compounds through different soil type. And as you can see in this bar chart, these compounds have strong ability or affinity to bind to the soil particles. Most of these compounds, except caffeine and cephalexin, which is we be able to recover almost just tiny percentage out of their spiked concentration in the soil column, as well as I mentioned in the previous, baclofen for the first time was studied its behavior through the soil. Next, please. Conclusion. The likelihood of groundwater to contamination by these selected compounds, especially the detected one, it's negligible due to high removal efficiency, as I mentioned in the previous slides, low percolation as well, and because of the groundwater in this area level is about 50 to 60 meters. So in the end, we can say the use of effluent for irrigation and groundwater recharge, it is unlikely to cause any environmental risk. Next, please. Thank you. Thank you for listening.
Thank you, Obaid. So our next presentation is from Maryna Strokal, working as assistant professor in Netherlands.
Thank you so much for this opportunity to give a talk today in this conference. I'm Maryna Strokal from Wageningen University from the Netherlands, and I will be talking about multiple pollutants in rivers from wastewater. And I will focus on synergies and trade-offs and future pollution control. So my talk will be about the future and the scenarios. Next slide, please. And in my talk, I will focus on three key messages for the 21st century. The first is that wastewater is expected to be a common source of multiple pollutants in rivers worldwide. Second, more sewage connections may result in more pollutants in many waters globally. That's what I consider as a trade-off between the sanitation intervention like sewage connections and water pollution. And the third is that better wastewater treatment may reduce pollutants in many rivers, except for Africa. And I will explain how I came to those messages in my talk. So the next slide, please. I'm showing you some pictures illustrating that rivers in the world, they have multiple pollutants. And often, actually, they are cocktails of emerging pollutants, and the previous talks also illustrated some examples and highlighted water pollution issues. And those cocktails of emerging pollutants, they have different impacts on nature and society. And the next slide, please. On this slide, you see a cartoon that summarizes the human activities on the land and the river in blue flowing into the coastal waters. And what we analyze in my group, that wastewater is expected to be a common source of multiple pollutants. And I'm highlighting that on the cartoon with the frame, the blue frame. And that's because in the 21st century, more than 2/3 of the global population is expected to live in urban areas, and that means more sewage connections. And if treatment is not proper, then more pollutants may enter rivers from the sewage systems. And pollutants, for example, pathogens that are in feces and urine, also nutrients. And among the emerging pollutants, microplastics from car tires, personal care products, and different chemicals, such as triclosan, which is antibacterial agent that is used often in soap when we wash our hands. And there are many other examples that the previous speakers also showed. The next slide, please. So we model the flows of pollutants from land activities to rivers and to the seas worldwide, and we use the Maryna model that stands for models to assess river inputs of pollutants to seas. I'm showing you a cartoon that summarizes which pollutants we consider in our models, and we integrate different modeling approaches to calculate the flows of pollutants from land to rivers and seas. So our models are evaluated for multiple pollutants. Important model inputs are population, wastewater treatment, sewage connections, and others. And the outputs are pollutants in rivers by source and subbasin. And that's the strength of our modeling approach, that we can tell or we can show where in the world from which sources pollutants are coming to water systems. And we do this for more than 10,000 river subbasins in the world. That is shown on the map. The next slide, please. So the Maryna models are used to better understand pollution hotspots and their sources and synergies and trade-offs in pollution control. And we do that through scenario analysis. I will show you a few examples. The next slide. So two examples, two scenarios, they are based on shared socioeconomic pathways, and those existing pathways that have been developed by other scientific communities, and we interpreted them for water pollution. So the two scenarios, the first one is economy first, based on the shared socioeconomic pathway 5. And the second is sustainability scenario based on the storylines of shared socioeconomic pathway 1. And these two scenarios, they have common storylines for population growth, which is expected to be low globally. Urbanization is expected to be high, so more people will live in cities in the future. And there will be a lot of sewage connections in those two scenarios. But the difference between them is the speed to implement advanced wastewater treatment technologies. In the economy first scenario, it's slow, so not much improvement. And in the sustainability scenarios, it is high, so a lot of improvement. The next slide, please. I'm showing you 5 maps for 5 pollutants in rivers. So these are the model results for sewage systems in the year 2010. You don't have to look into details of those maps. You need to look at the color codes. So there are 5 maps for one for nitrogen, phosphorus, microplastics, triclosan, and cryptosporidium, which is a pathogen microorganism that can cause diarrhea. And the colors on the maps range from light to dark blue, and dark blue are high levels of pollution. And we calculate multi-pollutant hotspots that are located in North America, Europe, and South Asia, in many subbasins there. And those are the areas where that cover 20% of the global land surface area. but those areas accommodate more than 2/3 of the global population. And that indicates that many people in the world actually live in basins that are contaminated with multiple pollutants. The next slide, please. I'm going to show you the results for the future trends, and first for the economy first scenario. The map shows the changes in river pollution during 2010 and 2050, and the color codes range from green, which means less pollution in the future, to dark red, which means that those are the basins that we calculate will receive more pollutants in the future from sewage systems. And you see a lot of red colors on the map for many basins in the world, and that indicates that in the economy first scenario with high urbanization, we expect more river pollution with multiple pollutants. And the next slide. So more sewage connection may result in more pollutants in many rivers worldwide. That's the conclusion that we derive from this modeling exercise for this scenario. And the next slide. Now the results for the sustainability scenario, and then click again. And the map, it is a similar map that I showed before, but for a different scenario, for the sustainability scenario. It shows also the changes in river pollution during 2010 and 2050, but the colors are now, many basins have green colors, which indicates that with improving wastewater treatment, rivers are expected to become less polluted from sewage systems. However, this is not the case for many basins in Africa. So for Africa, it will be challenging to reach pollution levels of 2010 in the future. And those are pollution levels associated with the sewage systems. And that is because of the very rapid increase in the population number. So more people, more waste, and this waste will go to the sewage systems, and it will be difficult to reduce pollution to the levels of 2010. And the next slide, please. So those are the three messages that I showed you at the beginning. And during my talk, I explained to you how I came to them. So the first one, that the wastewater is expected to be a common source of multiple pollutants in rivers. The second, that more sewage connections may result in more pollution if treatment is poor. And the third is that better wastewater treatment may reduce pollutants in many rivers worldwide, except for Africa. And my last slide. I thank you for your attention, and also would like to acknowledge the effort of my team who develop the models, and with those models we can use and learn about the pollution hotspots and synergies and trade-offs in pollution control. Thank you for your attention.
Thank you, Maryna. So our next presentation will be from Yunus Ahmad, affiliated to Australian Centre for Water and Environmental Biotechnology, University of Queensland, Department of Chemistry, Chittagong University of Engineering and Technology. And the presentation will be on removal of emerging pollutants from urban water, alternation of existing tertiary treatment process by using heterogeneous photopentant process.
Hello, everyone. Hope all are good and enjoying the conference. Yes. I am Dr. Yunus Ahmed from Bangladesh at this moment. Today conference, I will present removal of emerging pollutant from urban water and alteration of existing treatment processes by using heterogeneous photopentant process. Why we need alteration for our existing process? I will explain it in the following slide. We know water is the driving force for all the nature, but global water scarcity. If we consider the alarm of global water scarcity, treated and reused wastewater would be the cost-effective, sustainable, and long-term solution. This is next slide. Yeah, this is the conventional wastewater treatment plant, existing process. But if we see this one, the existing treatment plant completely unable to remove ARB, ARG, and micropollutant. And their next slide, yeah. And their performance was varied. Average 3 log ARB removal efficiency and 61% micropollutant removal efficiency observed on this existing treatment process. Next slide. What happened if not fully treated our existing treatment process? If ARB were not killed completely, the residual ARB could spread the antibiotic resistance, not only by vertical gene transfer, but also through the horizontal gene transfer as the donor. If ARG are not damaged completely, the residual ARG could be uptaken by a recipient through transformation, thus resulting the dissemination of antibiotic resistance too. If micropollutant were not degraded completely, it will affect the aquatic environment by increasing toxicity, endocrine disruption, antibiotic resistance development in the microorganism, as well as human chronic diseases. Next, please. So, which process do we expect? We expect one-step process. What is one-step process? One-step process is not only kill the ARB, but also degrade ARG and micropollutant completely. How can we get this one? We need to develop the process. This process just create sufficient reactive oxygen species. This reactive oxygen species is possible to damage the bacterial cell membrane and then degrade the DNA. And also, if we see here, it's possible to degrade the ERG and also micropollutant. And last, it's produce the mineralization product. So, next. Now, I will show here my research outcome, because this is totally, I will show here my PhD outcome. In this slide, I will show my 3 articles. These 3 articles, it is totally covered this photoventon process, different photoventon process. In the first article, I was published in Water Research. It is my homogeneous photoventon process. In this whole photoventon process, it was a proof of concept. In this process, we just remove, we are able to remove 6 log ARB and ERG. But problem is here, it's take higher treatment time and doses for IRG than ARB. But faster degradation of long amplicon ARG and short amplicon if I compare. And the second article I also published in Water Research. It was my modified photoventon process. In this modified photoventon process, I using EDDS to control my pH in the reaction medium. In this process, I will be able to remove ARB, ARG, micropollutant simultaneously, and also possible 6 log ARB reduction. Extra ERG also possible within 10 times, and I able to 5 representative micropollutant I remove and degrade up to 90%, 99%. Second, next slide, please. Yeah, this is my final outcome of this process. It's a heterogeneous photoventon process. In this heterogeneous process, I able to remove ARB, ARG, and micropollutant also say more than 6 log ARB and ERG completely. This is the heterogeneous process. We know homogeneous process have some limitation. It is unable to reuse this process, this catalyst, but heterogeneous process we are able to remove this one. And it's this nanocatalyst, it's a photocatalytic and good recycle ability. The proposed process is optimistic one-step process. So we can think this is the alternation process for our existing treatment disinfection process. If we develop this process more, then it will be the good alternative to remove ARB, ARG, and micropollutant easily. Next, please. Now, in this slide, I will acknowledge my supervisor and also my Australian Government Research Council for giving me RTP scholarship. Then I finish my PhD here, and now I am doing here job. Thank you.
Thank you, Yunus. So we'll go to the next one, which is the final presentation. Yayan Huang from China, Research Center for Eco-Environmental Sciences, Chinese Academy of Sciences. And The topic of presentation will be on facilitated prediction of emerging pollutant degradation by UV AOPs in practical waters through combination of models, simulation, and portable.
Good morning, everyone. My topic is emerging pollutant degradation by UVLPs in practical waters. Next slide, please. As we know, emerging pollutants nowadays represent a key scientific and technical challenge to water safety. PPCPs and EDCs are two typical groups. Each group contains various subgroups. Therefore, there are hundreds or even thousands of emerging pollutants in our environment. Next, please. Next slide. UV-based advanced oxidation processes called UVLPs have been proved highly effective for the removal of emerging pollutants in water. It has been widely used in the advanced treatment of drinking water, sewage, and reclaimed water all over the world. Next slide, please. The performance of UVLPs to remove the emerging pollutants are affected by many factors, such as different light sources. It includes low pressure, medium pressure mercury lamps, various UVLPs such as UV persulfate, UV chlorine. In addition, different water such as drinking water and industry wastewater have different metrics which compete for reactive radicals at different degrees against the target emerging pollutants. Therefore, considering the different types of these conditions, the number of experiments seems infinite. Next, please. Conventionally, if you want to evaluate the removal efficiency of target emerging pollutant by a UVLP process, you need to collect the test water on site and ship it to your laboratory, conduct experiments using a bench photoreactor, analyze the target emerging pollutant using advanced instruments, and then evaluate the removal efficiency and the treatment cost. This means a very heavy workload and high professional requirements, which is very time consuming and expensive. Therefore, it is urgent to develop a rapid and accurate method for UVLP performance evaluation. Next, please. We have developed a model simulation combined portable measurement method. The model simulation consisted of 3 models. The first one, the steady-state reactive radical model. It represents the balance between the formation and the consumption of reactive radicals, as shown by these 2 equations. The second one, the quantitative structure activity relationship model, called the QSA model. It can predict the second order reaction rate constant between reactive radical and target emerging pollutant. The third one, the photochemical model. It can calculate the formation rate of the reactive radical in UVLPs. Portable measurement was conducted on a mini fluidic photoreaction system. It could measure the reactive radical scavenging capacity, the RRSC of water matrix. This parameter represents the capacity of water matrix to compete for the reactive radicals against the emerging pollutant. The procedures are as follows. Spike the probe MB methylene blue into different concentration surrogate matrix solutions. Then a standard curve could be obtained. Then spike the probe into our test water. Then the target RSC of the test water could be obtained. Combining these parameters, the UVLP performance could be predicted. Next, please. 7 test water from different treatment processes in different water treatment plants in China were collected, and there are some water quality parameters. Next, please. Here are the results and the discussion. 3 QSAR equations were established. Each equation involves 2 important descriptors. The figures showed that the predicted second order reaction rate constant agreed quite well with the literature ones. It proved that the accuracy of our established QSAR model. Next, please. 3 standard curves of RSC were plotted, and 8 test waters, including ultra-pure water, were tested. Here, the finger showed the RSC values. As we expected, the more complex the water matrix is, the higher the RSC value. The ultra-pure water had the lowest RSC value. The primary sedimentation tank effluent in a sewage treatment plant had the largest RSC value. Next, please. We select the sulfate method, caffeine, carbamazepine as the 3 model micropollutants and predict the degradation rate by UVLPs using MSPM method. These figures showed that the predicted results using MSPM method agreed quite well with the experimental results. Next, please. Here are the conclusions. 3 parameters were determined by 3 models, and the RSC values were determined by portable measurement. Combined these two parts, MSPM method was developed to evaluate the UVLP performance rapidly. A paper was published in Water Research previously, and based on this method, we developed an MSPM package, which can be easily carried in the field to for the on-site evaluation of UVLP performance. And software was under development to predict the degradation rate of emerging pollutants by UVLPs. Next slide, please. Thank you for your attention.
Thank you, Ian.
Okay.
So thank you. We have come to the end of this session. I thank all the presenters of this session. We want to remind you that you can see an excellent selection of submitted conference posters at the conference website at www.iwraonlineconference.org. These posters can be found under the posters menu. And if you have any questions on these posters, you can send us an email to online.conference@iwra.org, and we'll contact the poster authors to respond to your queries. With regards to the PowerPoint presentations you have just seen, they'll also be made available soon on our conference website, along with the video recording of this session. And selected papers presented in the conference will be published in journal, and those authors will be contacted in near future. So I'm handing over Dahlia for the management of Q&A section.
Thank you, Surya. Actually, we got a lot of questions, more than 60 questions. So, but most of them were answered in the Q&A box. I have one question. I think it's for Obaid, that regarding his presentation, and climate change has been taken into account in your research or not?
No, actually, we didn't study that, unfortunately. So maybe this it's a kind of recommendation for the next future, near future research. Yeah.
Thank you, Obaid. I think all the questions have been answered in the Q&A box, so we're fine here. So back to you, Surya.
So I also don't think any of the questions are not answered. Most of them answered. And if further any questions are there, and they'll be answered, and the answers will be put into the conference website. And is there any questions from panelists? Okay, so I again thank you all the participants, the panelists and officials for joining this conference and joining this session, and to make a fruitful session. And all the PowerPoint presentations will be made available on the conference website soon. And do also see the posters which are available on the conference website. I thank all of them. Back to IWRA.